Hello class, today we are going to learn about the B cells and the humoral immunity. Each B cell carries immunoglobulins on its surface. The majority are IgM and IgD, all of which are specific for recognition of the same epitope. B cells selected to mature can be activated. This allows activated B cells to produce plasma cells and make antibodies as well as memory cells. This process is called clonal expansion or proliferation. Clonal selection includes stem cells differentiate into mature B cells, each bearing surface immunoglobulin against the specific antigen. B cell class II encounters its specific antigen and proliferates. Some B cells proliferate into a long-life memory cells, which at a later date can be stimulated to become antibody-producing plasma cells. Other B cells proliferate into antibody-producing plasma cells. Plasma cells secrete antibodies into circulation. B cells require help of T cells for most protein antigens. B cell internalize antigen and present it to T helper cell in combination with MHC class II molecules. If T cell recognizes antigen, it activates B cell. When an antigen-presenting cell makes contact with an antigen that can combine with its particular receptor, the APC, and the antigen bind. The APC then internalizes the antigen and digests it. Next, the APC displays digested antigen fragments on its surface by combining them with its major histocompatibility complex, MHC. T-independent antigens have repeating units, epitopes, that can cross-link several antigen receptors on the same B cell. These antigens stimulate the B cell to make antibodies without the aid of T helper cells. The polysaccharides of bacterial capsules are examples of this type of antigen. The human immune system is capable of creating a mind-boggling number of antibodies, an estimated minimum of quadrillion of them. The number of genes required for this account of diversity is actually relatively small, thanks to random rearrangement of gene segments that code for antigen receptors, resulting in variations of the amino acid sequence at the antigen binding site. These rearrangements occur before antigen is present, during the early stages of the differentiation of each B cell. When an antibody encounters an antigen for which it is specific, their binding forms of an antigen, antibody complex. The strength of the bond between antigen and antibody is called affinity. In general, the closer the physical fit between antigen and antibody, the higher the affinity. Specificity. Antibody recognizes a specific epitope. The antibody molecule itself is not damaging to the antigen. Rather, the binding marks foreign cells and the molecules for destruction and neutralization by phagocytes and the complement. Foreign organisms and the toxin are rendered harmless by several mechanisms, as summarized. These are agglutination, opsonization, neutralization, antibody-dependent cell-mediated cytotoxicity, and activation of complement. The binding of antibodies to antigens form antigen-antibody complexes that tag foreign cells and molecules for destruction by phagocytes and the complement. Overview of the complement pathways. All three pathways have different triggers and starting points, but they all converge at the same place, C3 convertase. This enzyme begins the series of reactions that give rise to the membrane attack complex characteristic of the component machinery. The final result of the formation of the tiny openings in the cell membrane and the destruction of the target pathogens. Thank you for your attention.